This is lecture 12. We're going to do the rest of the discussion of worms. One of the main focus of this lecture is going to be a series of signals that come from the P2 cell and basically set up the dorsal ventral axis. We'll talk really quickly about We'll talk really quickly about left-right specification, very briefly because it's not very well understood, but it's kind of interesting. And then we'll talk a little bit about the development of the vulva of the worm, which happens much later in this process. It is in the early instar larvae that this takes place, but it's a very well understood model of how organogenesis takes place, and we don't spend much time talking about the formation of specific organs throughout the semester because we haven't got time to go into that level of detail, but it's a really good simple example of how several different types of cells develop to form one particular structure. And we'll just look at, there's many genes associated with it, but we're just going to look at 3 of them. So I'm trying to keep the number of genes to memorize to a minimum. But that being said, I will then be asking a lot of, what happens if we express this gene here instead of there, or what happens if we shut it off, and that sort of thing. So there'll be more of that and less of just memorization. Okay, so where we left it last time is we had talked about the anterior-posterior axis and how an asymmetric cell division, both in size and in the determinants present inside the cells, gave rise to an anterior cell called the AB cell and the P1 cell. And the AB cell, so the second division that takes place is not synchronous. It doesn't happen at the same time. And the egg is not perfectly round. It's narrower at one end than the other. And what's strange is it's narrower at the end where the larger cell is. So what happens is when that cell starts to divide, because of, I think, just the physical constraints of being in a more oval type of egg, it causes the plane of division to be tilted relative to the previous one. So the previous one was like this, and this plane of division is at an angle to it, not quite 45 degrees, or well, maybe almost 45 degrees. And so when they divide, there is a cell towards the more posterior called the ABP. There's a very precise way of naming all the cells, and a more anterior one called ABA. Okay, and so this part here is just for your reference. It just shows sort of the gradual alteration of the plane of division. The cell is starting to divide, but it gets squished off to one side, and then we wind up with the P and the A cells. After that, if we think about those planes of division, then the P1 cell divides and it creates a P2 cell, which is smaller and towards the posterior, and then a larger cell, which is the EMS cell, and this is the ventral specific cell. And so therefore, because this is ventral, that then automatically means this is called the ABP cell, and it does have a more posterior fate than this one. So of these 2 cells, this is posterior, but also it is on the dorsal side of the embryo. Okay, and so the 2 key things are the divisions are asynchronous. They don't have the same plane of division, and there is something about the physicality of having an egg that's a little narrower at one end and wider at another that causes this sort of odd offsetting of the division plane. Okay, and then it turns out that the P1 and the P2 cell, P1 before it divides and then P2 afterwards, it's a source of signals to the ABP and the EMS cells. And ABA and ABP are genetically you know, in terms of what they're actually expressing at this point, they are the same, but only one of them is in contact with the P2 cell. And so if you were to take a needle and just kind of shove these around a little bit at this stage, 10 minutes, up to 10 minutes after they have formed, you shift them around, you can make them trade fates. But if you wait until later, you can't. And so that tells us that there is a time-limited signaling or information from the P2 cell that sets the fate of this cell. And you can actually change it if you trade the positions of those two cells, but only within a certain time window. This is interesting because this is one of those organisms where we think of the development as almost entirely mosaic. It's almost entirely like pre-specified and set up, and every cell we know exactly what it's going to form, and if you zap that cell with a laser, all the derivatives of that cell don't develop. So under normal circumstances, you don't see a lot of evidence of sort of what we would call regulation adjustment, but you can make these experimental manipulations, and if you do them within the right time window, because it is signaling and communication between the cells that is still leading the development, causing the development to occur as it does, you can make some alterations in the cells. And that's just a really interesting point to keep in mind. Okay, so this is one of the, an example. So normally we've got our AB and our P1 cell. The AB cell divides to form the ABA and the ABP cell first. And when that happens, this cell, the P cell, has a lot more contact with the P1 cell. And then after this cell starts to divide, the ABP is the only cell that has contact with P2. So right after these form, and before P2 has formed, there is a way to, you can't just move cells around willy-nilly, but if you take a glass pipette that's not sharp, you don't want to poke or damage anything, but you can actually take and put pressure on the anterior part of this and just kind of push it. And if you do that, you shove it so that the ABA cell, really, the one that was going to be the ABA cell, is actually more towards the posterior, and then the P cell is shifted towards the anterior with this manipulation. And then if you watch then the development afterwards, then the A cell is down here, but it's actually going to adopt the ABP fate. And this one is going to adopt the ABA fate in terms of the structures and things that it produces, this being the dorsal side, and the EMS cell develops on top instead of on the bottom, and that sets the ventral. So what you have here, the worm is still normal, and its left and right is still set up. It's opposite though. Notice that it's opposite to this, because when the worm is upside down, then left and right is on the other sides too. Think of your own arms when you're lying on your back versus if you were lying on your front. And so you think, oh no, this worm is upside down. But once it hatches out of the egg, it's perfectly fine, right? There's not going to be any problem. So during development, the poles are reversed here, but it doesn't have any consequence to the living organism. But it's interesting to watch because things are sort of appearing in the wrong places, but then, you know, because the worm is only upside down, it's not a problem. Okay, so I want to talk about one of the first signals between the P2 cell and the ABA and ABP. And that involves GLP1, which is the Notch gene, the Notch receptor. Early in development, when the egg is first developed, the RNA for this receptor is present throughout the embryo, but the translation is repressed in the posterior. So translational repression in part of the embryo. This should sound familiar to you. Think back to where we've seen it before. When we study development, sometimes organisms are quite different and it's hard to find the connections between them. So I'm going to try to remember to point out whenever there is one so that you can think back to the other topics that we've already covered and see how there are these connections, how certain mechanisms are seen in lots of different organisms. So after the first division, that asymmetric division, remember, the P1 cell has P granules and things like that in it, but it doesn't have any of the GLP-1 receptor, so the AB cell has it. After that first division, you've got two cells and they both have the GLP-1 receptor, so they still both have Notch. And the P1 cell is expressing the ligand for that, which is called ASX1, but it's basically delta, right? So delta and Notch, they were named in flies because of the wing phenotypes associated with them. And it turns out there's multiple notches and deltas in lots of different organisms. So we've got a ligand for a receptor, and we've got one cell in much stronger contact with P1. And then after division to make P2 and EMS, only one of these cells has contact with the cell that is expressing the ligand. Okay, and so we start off with equivalent cells, but now this one has received signals and information that this one has not, and that's the key to how this process works. So part one is the APX1 ligand being expressed on the P2 cell, and the ABP cell having the receptor for it, receiving the information. ABA also has GLP-1, but it's not touching the P2 cell. So think about why it matters that these two cells are touching each other. Okay. And so this cell receives different information and develops as ABP rather than ABA. Okay. And there's figure 6.9 in the text, which I'll probably show in lecture, but it's basically the same information. And sometimes when you see slightly different figures, they are helpful. Okay. So that's part one. Part two is that after P2 and EMS cells are formed, P2 is expressing MOM1, which turns out to be wingless. Again, wingless was named in flies, and it has to do with a role in wing development. Frizzled as well, named for a wing development phenotype. But in worms, it's called MOM2 because it was named after a worm phenotype before it was known that it was actually wingless or wint. Wint is the name in mammals and other vertebrates. And so there's a MOM2 signal, short-range diffusible signal. MOM5 is the receptor for that, frizzled. And when the EMS cell receives the signal from P2, there's strong signal close by. It's short range. It doesn't travel all over the place. So when this cell then divides, the second messengers from the Wnt signaling, they're mainly on this side. And so after the cell divides, the one that had been receiving really strong MOM2 Wnt signaling develops as the E cell, which forms endoderm, the gut. And the other cell, in the absence of that signaling, or with very low levels of it, not enough to initiate transcription of the endoderm specific genes, will then go on and form the default, which is mesoderm specific. Yeah, and so this is you will see Wnt signaling over and over again in a number of different organisms when we talk about mouse and frog and that sort of thing. But just to note that the system is a little bit different in nematodes, and so I'm going to talk a little bit about that signal. So when the cell receives the MOM2 signal through the frizzled, there's a receptor on the surface here, frizzled, and then there's downstream activation. And basically there are two proteins that are similar to beta-catenin. They are the worm forms of beta-catenin. One is called cis1 and one is called worm1. And worm1 is in a complex with lit1, and cis1 is alone in the cytoplasm. So you've got these two forms of beta-catenin. They're not identical to each other, and they do slightly different things. So when cis1 is stabilized, so there's no destruction complex, and the beta-catenin here, cis1 is normal, it goes into the nucleus and it can act as a cofactor with POP1, which is a transcription factor, and it activates endoderm-specific genes. In the absence of worm1 and lit1, these two together, this is a kinase, and this is a beta-catenin, as I said. They can enter the nucleus, and what they do is they actually are able to phosphorylate POP1, the transcription factor. And when they do that, it's in huge concentration in the nucleus in the absence of Wnt signaling. And when there's lots of it, it kind of loads onto the regulatory sequences of the genes, and it turns them off. It prevents them from being transcribed because it's forming these complexes, multi-unit complexes with itself, right? But when the the lit1 is brought in, and worm1 binds here, and the lit1 does the phosphorylation then of POP1, basically it takes a lot of it outside of the nucleus, and then when POP1 is in smaller concentration and bound to cis1, they are able together to transcribe the endoderm-specific genes. Okay, not transcribe, but activate it, because of course RNA polymerase does the transcription. So this is a kind of a weird system because there's two different things happening. One is to release some of the POP1 out of the nucleus in order to allow the smaller concentration you have to bind with cis1 and then form this complex that activates transcription. And the other, having these two different beta-catenins that do kind of somewhat different things is also sort of an interesting feature. Okay. And I think I'm going to leave out the left and right just for this prep, because it's not really that crucial. Just the idea that the plane of cleavage of the third division determines the left-right axis, and you can actually make some manipulations to that and cause the left-right to switch. And I'll talk about it very briefly in the lecture, I think, rather than go into a huge amount of detail, because I want to make sure I spend a little bit of time on the vulval development. As I said, the vulva is a model for organ formation, and it's not a topic in general that we'll be able to spend much time on. And this is a relatively simple one, so it won't take much of our time to talk about it. So it's a good chance to discuss. And so the formation of the vulva is rather an important structure because worms are hermaphrodites. They can fertilize their own eggs, and so you have to extrude those eggs out of the body, and if you don't have a vulva, you can't do it. So I remember I was a grad student when they started identifying these bag of worms genotypes where there was no vulva development. They were looking for other things, but they found this as one of the mutant phenotypes in the screens. But you can also have mutations in genes that cause too many vulvas. So this shouldn't say too many mouths, which is a different thing. It's a multi-vulva phenotype. Too many mouths was just a kind of a nickname, sort of slang, which we shouldn't be using. We should use the correct term here. So multivulva. So what happens is you're missing some of the epidermis, which is called hypoderm in the worm, and you wind up with like a whole lot of vulva. I've seen pictures where there's 6 different ones, for example, on the worms. And so we can find out, having screened for these, we can do some work to find out which genes are responsible for these phenotypes. So this is the basic process. So here's our first instar worm. There's a gonad, and within the gonad, there is an anchor cell, which I should just move this over a little bit to make it really clear. And this anchor cell is the source of signals. And basically, early in development, there are 6 cells, and this is their names: P3, P, 4, 5, 6, etc., up to 8. These 6 are set aside, and they're not able to form hypodermis, or the skin of the worm. Okay, initially they're not. And then the anchor cell sends signals, and it ends up influencing the 3 closest cells to itself, which end up forming the vulva. These 3, they combine. So the primary cells is sort of the actual opening and immediate surroundings, and then the secondary cells form sort of supportive structure. So these together form the vulva, and then eventually, after a few steps in development, these guys that were not allowed, that were restricted, prevented from forming hypodermis to begin with, then go ahead and form it after all. So they're able to go on and form hypodermis, which is just regular epidermis. And so this is, I grabbed this from another image, because there's one in your text, which when I look at it, I just go, I think I would rather not try to learn from. I find it confusing, and it's possible you can look at it, of course, yourself and see if it's helpful. For me, this is a more helpful one. So here's our anchor cell and the gonad. These are our vulval precursor cells. So there's 6 of them, but only 3 of them are actually going to form the vulva. 2 that form secondary, which is kind of the supportive tissue, it's shown here in side view, and then 1 that forms the actual opening and a slight sort of invagination. And this is where the eggs are going to be extruded from. And then the tertiary ones, are the ones that are going to go ahead and form the actual non-vulval tissue, which is hypodermis in this case. So primary, secondary, and tertiary. And then these are the signals. There's a whole bunch of additional ones, but I think this is more important, is to just get a few basics right. So the anchor cell sends a signal, which is Lin3. It's epidermal growth factor. There's lots of different EGFs as well. This is one of them, and Lin3 is the name of the one that the anchor cell secretes, and it's a short-range diffusible signal. So the vast majority of it goes straight across to the P6P cell. And then there's a lesser amount to the P5P and the P7P cell. Okay, so all of these guys have been told, okay, you are going to form the vulva. But what happens is, and LET23, which is short for lethal 23, they're named after lineage and lethal and stuff because they didn't know what the names of the actual proteins were, right, at the time. So it's hard to remember all the names, but I'm going to try to, that's why I'm keeping them to a minimum. So you've got your EGF receptor receiving, and the receptors, all these guys have receptors too, but they don't receive any of the signal, right? So these guys go ahead and form the tertiary fate. But the cell that received the strongest amount of Lin3 then starts to send an inhibitory signal, and it's lateral inhibition. And we've seen this before. Think of bristles in flies that I talked about, I think it was in lecture 3. And so this signal is going to be delta ligand, and then this is another kind of notch called Lin12, and this notch, when it's received, it has an inhibitory effect. So this signal goes, okay, you can't be primary cells, so you're going to have to adopt with your small amount of Lin3 signal and plus my lateral signal. That means you're going to have to form the secondary fate, and you too on this side, you're going to have to form that fate as well. So we've got a strong Lin3 for the primary, and then the primary cell sends out an inhibitory signal to the cells to either side of it, and these guys have a little bit of the Lin3 plus the inhibitory signal, and then that gives them their secondary cell fate, and so they form those supportive structures. While these guys, there's going to be 8 of these, and I think 6 each of these guys, or maybe it's 8 each. I can't remember. But each of them forms multiple cells, and they form overall a 22 cell structure, which is the vulva with the vulva proper, the primary cells, and then the supportive cells, without which the muscles to extrude the eggs probably don't function properly. So for a functional structure, you have to have both of these types of cells. And then just imagine, so if you look at this diagram, think what happens if these guys don't have the receptor, the LET3 receptor. So if the worm is mutant for LET3, it means there's no receptors on any of these cells. So then what happens? Or what if there's no Lin12? What if these guys can't receive the inhibitory structure signal, and that sort of thing? So think about that, because I'll probably ask something in the class. Okay, and I'm just going to erase all this mess. And so I think that's probably enough information at this point, just to understand this this process in that much detail. That's enough. Next time we're going to look at sea urchins, and sea urchins are pretty cool. They are actually closer related to us than worms, evolutionarily, and we'll talk more about them next time.